Welcome back students to our new video. Today we are going to study about the type of immunoglobulin, and the first types of immunoglobulin we are going to study is the IgG or immunoglobulin gamma. Let's see about the immunoglobulin G or gamma immunoglobulin. So first before studying the about the IgG, I am going to explain about the classes of immunoglobulin. So antibody can come in different varieties are known as isotypes, and these isotypes in placental mammal there are five types. There are five types of antibodies or five types of immunoglobulin. These are IgA, which is Ig alpha, IgD, which is Ig delta, IgE epsilon, IgG gamma, and IgM, which is mu or immunoglobulin mu, and which are further subdivided into like IgA1, A2, A3, IgG1, G2, G3, like they are in numbers. And the prefix Ig stands for the immunoglobulin, and while the suffix denotes the heavy chain of the antibody. So the heavy chain of we study in earlier video we study about the structure of antibody. So antibody consists of one light chain and one heavy chain. So the heavy chain depends upon the consist of the various uh, alpha region, gamma region, delta region, epsilon region, and on the basis of this heavy chain the immunoglobulin is named, which is IgG, A, D, and etc. So the distinctive feature of each class are determined the part of the heavy chain within the hinge and FC region. FC region is the crystallizable fragment which present on the constant region, and hinge region we already know that it is the movable part or the flexible region which help in holding the structure of the antibody and also helping in attaching to the antigen. Let's see about the IgG. The immunoglobulin G or gamma immunoglobulin has a molecular weight of 1,50,000 Dalton, and the heavy chain type is 53,000 Dalton. The IgG or gamma immunoglobulin is the most abundant class of immunoglobulin in serum and constitute of about 80% of the total serum immunoglobulin. So students, remember that the in which immunoglobulin is very high in number, IgG is very high in number, and this IgG is made up of 2 kappa and 2 lambda chains. This immunoglobulin G or the other immunoglobulin also contain the 2 kappa or 1 lambda, but in IgG it mainly consists of 2 kappa and 2 lambda light chains. This is on the light chain. Remember student, this is on the light chain, and this IgG is another then subdivided into IgG1, G2, G3 and G4. It has 4 subtypes and on the basis of decreasing serum level. So why on the basis of decreasing serum level students? Because the number of IgG1 is high, then number of IgG2 is low, then on the basis of the decreasing number of the immunoglobulin G subtypes in the serum level, so according to them these subtypes are divided. The highest number is Ig1 or the lowest is Ig4, or it vary in person to person or animal to animal. So it has the longest half-life among the other antibody. The half-life of the immunoglobulin G is about 23 days. Imagine student that after an immune response by an antigen, the T helper cell activate the B cell, and this B cell produce a large amount of immunoglobulin which is IgG, and this IgG has a shelf life of 23 days. So if you have any infection and you recover from that infection, but after even that immunoglobulin or the antibodies present in our body is about 23 days, and that immunoglobulin is the IgG, and this IgG is the only antibody. Remember student, it is a very important point of the IgG is that this is the only antibody which can be transferred from the placenta. It can cross placenta. No other antibody can cross placenta, because if any other body cross the placenta, the immune response of the baby can also trigger the against that immunoglobulin. But this IgG when transfer or cross from the placenta, the baby immune system take it as a self, And mother immune system also, if any transfer from that baby to mother is there, it also take it itself. So remember student that we all that IgG present in us is from our mother. Let us consider that, or you can take it as a consideration, because this IgG can cross placenta. So if we got the IgGs from our mother, and this immunity is known as the natural passive immunity. And we all know about the active immunization, passive immunization, and it is a type of natural passive immunity. And IgG also activate the complement system. Even it is a complement activation like alternate pathway or classical pathway or other different pathways. So IgG play a very vital role against the infections. So what types of infection IgG can be secreted, or in against which infection IgG is secreted? Let's see. So the biological functions of IgG. IgG is the major antibody producing secondary immune response. There are two type of immune responses, primary and secondary. So the IgG are produced in high number again in the secondary immune response. The IgG3 and IgG4, so these are the two subtypes. IgG3 and 4 can cross the placenta. So you have to remember student, Ig3 and Ig4 can cross placenta and play an important role in protecting the fetus. So these cross placenta, and these are the mother's immunoglobulin, and enter in the fetus body and protect it from the other infection. The IgG3 is the most effective complement activator. IgG3 is play a very important role in complement activation in it, and it is a very effective complement activator. And this is followed by Ig1 and Ig2, but Ig4 not is a complement activator. Ig1 and Ig3 binds with a high affinity to the Fc region and help in the receptor to the phagocytosis. And we all know in the earlier videos, and we also studied about the antibody. So the Fc region, the effector cells like macrophages or natural killer cell bind to the Fc region, and the opsonization process is done, from where the phagocytosis is done for that foreign material or antigen. So IgG1 and Ig3 can bind to the Fc region. So you have to remember this point also. And IgG help in the bacterial immobilization, and it also neutralize the toxins and viruses. So IgG play a very important role in neutralization of the toxin and viruses, get rid of the viruses. So this is the structure of the IgG, or it is a monovalent in nature. So remember student, IgG is monovalent in nature, and it is the same structure like of our normal antibody. But other immunoglobulin like IgA is divalent, and pentavalent immunoglobulins are also there. So we are going to discuss it in our next lecture. Every immunoglobulin has its own lecture. So if you like that video, you have to subscribe, and thank you for watching. And if you have any doubt or difficulty, you can comment in the comment box. So I hope you like that video and you understand this IgG very easily. So thank you for your support. Thank you.